All right. So only a little bit more slides to sit through, and then I am unleashing you on a real-world data set from a clinical lab. So at this point, hopefully you are convinced that calibrating and validating is something you should be considering in your targeted mass spec proteomics methods. And what I want to do now is dig a little bit deeper into these 5 experiments proposed by the CPTAC working group. So the working group, after this paper that Sue was a lead author on, that summarized all of the results that they found, they sat down and they tried to synthesize kind of a summary of here are what we think are the most important things you should be doing if you're trying to do this kind of validated experiment. Before I dig in, I want to get a little bit on a soapbox and just tell you that we're emphasizing that you can't do quantitative proteomics without doing these calibrations and validations. And what we're doing is teaching you what worked for us. If Russ Grant or if Andy Hufnagel came up here and wanted to present, they might present slightly different opinions on this. I want to emphasize to you that not everyone in the field agrees. So I presented in the morning section, I showed you there was this Paradise Lost and Found paper that presented their viewpoints on this. I presented the Carr et al. Paper that emphasized their fit-for-purpose approach. Then there's this CPTAC working group proposed approach. So not everyone agrees on the specifics, but everyone agrees calibrate and validate. So you may hear contradictory things from either us 3 instructors at this class here, or if you go out to your own lab, maybe your boss will be like, yeah, I don't like to do my calibration and validation that way. It is up to you as an independent researcher, an independent scientist, to decide what you need to do so that it is all in service of meaningful and reproducible science. So Sue tried to give us kind of a reminder that what we're doing a lot of times with targeted quantitative proteomics by mass spectrometry is find biomarkers, discover biomarkers, validate biomarkers, health and disease, understanding biological processes. I've seen posters up here with Alzheimer's and with cancer, and we're really trying to make sure that what we're measuring and the conclusions we're making from it are meaningful and reproducible. So I showed you there's a lot of different opinions. Those were the 2 papers I had talked about before. There's this tiered method that came out from the Carr working group that recommended the 3 different tiers. There was the Grant and Hufnagel minimal set of experiments that they recommend for quantification. And then finally, what we're focusing on right now is kind of the 5 tiers of CPTAC, because I feel like they have poured so much effort into making this understandable that it's a good one to teach. So right off the bat, experiment 1 is the response curve. And we're going to skip that because we have an entire afternoon dedicated to the response curve because it is that important. The response curve is experiment 1. It's the most important, so we're going to spend a whole afternoon really, really digging in deep for response curves. So experiment 2 is what the CPTAC working group calls a mini validation of repeatability. So you may notice that these boxes are in different colors. So the mini validation is supposed to give you an overview, give you kind of a bird's eye view of different sources of variation that you might find in your experiment so that you can decide as a researcher, and you know the most about your targets, you know the most about your method, you can decide if you even want or need to go on to the more in-depth validation protocols. So this mini validation tries to encompass a couple different ideas that experiments 3, 4, and 5 dig deeper into. So experiment 2, the way you would set up this experiment is that you would take your stock solution and you would try to kind of get an idea of what is a quantitative value that I think would be on the low end, and to figure out what might be on your low end, maybe you go to literature and see if anybody else has measured your target, and if they have, what did they detect it at? What concentrations did they detect it at? You could also have some sort of biological intuition, like, oh, I'm going after a transcription factor. That might be really hard to quantify. Maybe I should just give up and change my thesis now. No, if you're doing transcription factors, you can still do it. I believe in you. You just need to enrich. You might, if you're doing something that's a follow-up on a pilot study, like Sue was doing with the CPTAC working group, maybe you have an idea of what range they were using before. So the idea here, and they write 350 x LLOQ, really the idea is low end and higher end. So I should definitely detect my 50x LLOQ. My 3x might be kind of hard to detect. It might be on the lower range. And then the CPTAC working group suggested the 80% ULOQ. Does anybody know what the U stands for? Upper limit of quant. And Sue had mentioned before, just to remind you, recall that she had mentioned the upper limit. A lot of times we don't worry about, because if your biomarker is so abundant that you will have an upper limit, you typically are golden and you don't have to worry about this. So maybe in a lot of experiments that I would do, I would suggest maybe not going after the upper limit and instead replacing that third sample with something that should just be screaming signal. Like, I will definitely detect this, it will be strong, it will be very easy to pick out in my data. And then you'll take these standards and spike them into your matrix. So I'm not sure if we've actually defined what we mean by matrix in this class. So what I mean is, if you are using an experimental system, whatever that system is, that should represent everything, every chemical, every interference that will be present in your sample. So as kind of an illustration, I work in yeast, in budding yeast. So my matrix would be a yeast lysate that has gone through my entire sample preparation process. So for me, that means I am bead beating to break them up mechanically. I'm to lyse them, so now I have a lysate, I reduce and I alkylate, and I digest, and then I desalt. So that desalted matrix is what I use if I were to spike some standards into a matrix. If you're working in plasma, human plasma, that would be plasma taken through your entire protocol. So it should be representative of whatever you would normally be putting on your mass spectrometer. So then when you go to analyze these three samples, you should analyze them, you should acquire them in replicates. So now you have a representation of a low detection, a medium detection, and a screaming signal detection, and you can calculate the percent coefficient of variation. And Sue will be taking us through doing this, and we'll be doing this a little bit in Skyline, but it just means how reproducible are those peaks. And from there, we can get an idea. So maybe we can tell at this point that one of these standards, as I measured it over time, the peaks got smaller and smaller and smaller. So if one of my samples is sitting in an autosampler on a liquid chromatography system, and the peak is getting smaller and smaller, that might indicate to me that something's wrong with my standard. It's degrading, or it's being modified, or it's sticking to the plastic on my tubes, and that might indicate to me I have a stability issue. So that's one of the tests later on, is stability. Yes?
So for replicate, just to be clear, you just mean a technical injection of the same sample?
In this experiment, we're just going to do a technical replicate. Yes. So we're just going to have our three samples, make technical replicates of it, stick them all in the autosampler at the same time, so they're sitting in the autosampler for the exact same time. We sample one, and then maybe a day or two later, however long it takes us with our method, we sample the next one. And what you're trying to get out of the data from this experiment is the intra and inter-assay variability. So the digestion kinetics, if you have a protein standard, might change if you have low versus high amounts of the protein in your matrix. You are using the lower limit of quant, LLOQ that you discovered in experiment 1 to assess the repeatability of measuring that multiple times. And then finally, because you're doing 3 replicates and waiting to measure them on separate days, you get an idea for how stable the standard is. And you're hoping that the standard is going to be representative of how stable your endogenous analyte is in your sample. So experiment 2 gave us kind of an overview of different problems our experiment might have. And for your experimental needs, maybe you're okay to stop at this point and say, I've assessed the variability, I've assessed the variability of my standard, of my quantitation, and how reproducible my assay is. And maybe you can stop there. But maybe you can't, because either you feel it is necessary to go farther or your boss tells you you have to. So experiment 3 starts to dig into the idea of selectivity. And if you're setting this up, you're going to take 6 representative matrices. So in my yeast example, I would take 1 batch of yeast and process it, and that would be 1. Another batch of yeast, process it, and that would be rep 2. So that should start taking into account the variability in how I'm processing my samples. If it's plasma, maybe this would be 6 individual patients. Maybe it would be 6 individual pools of patients to get an idea of different matrix effects that you might see in your mass spectrometry. So you'll take your 6 biological replicates of matrix, spike your standards. Again, we're going to kind of use a mini curve method of doing this. So there'll be a blank where we don't add any of our standards, some middle range set of standards that we feel pretty confident we'll see, and then finally a pretty screaming concentration of standards that we should definitely see. And then we go and measure this, and what we're trying to ask from this set of data is how does the response of our peptide target, our analyte target, change across these 6 different matrices. So coming up with some totally random example on the top of my head, maybe in your 6 pools of plasma, there's different representatives of different races. Maybe some race has an interference in that peptide that now you see, oh, this transition is being interfered with in one of my biological matrices because of any number of reasons that people's plasma might be different. Maybe diet, maybe someone in that sample had some dietary medical issue that they have an interference in that transition now. You've assessed that in this selectivity experiment, so maybe you remove that transition, or maybe you have to look for a different peptide because now you realize, I don't know, worst case scenario, maybe you didn't realize there was amino acid variant in some of your patients. And now that you've done this experiment, you've noticed, oh, this peptide is being interfered with, or is being contributed from a different amino acid variant. Experiment 4 gets more into depth into stability. So this is, I don't want to say it's the easiest experiment to do, but is, I think, the least work intensive to do. And what you do here is you're just going to make multiple aliquots of 1 sample, make 1 big batch of sample, aliquot it out, and then 1 set of replicates will be placed in the autosampler and run at time 0, wait 6 hours, run it at 6 hours in the autosampler, then run it at 24 hours in the autosampler. So that's assessing if I load up my autosampler and press start on my queue and walk away for a day, are the peptides still going to be there when I get there in the morning? The other set of replicates are placed in the freezer to test freezer stability. 1 is frozen for 6 hours, and then those are measured on the mass spectrometer. Another one is exposed to a freeze thaw. And the last one is stored long term in the freezer for 4 weeks to see if any potential modifications happen or if you're losing things to the sides of, to the plastic on the sides of your tubes. So I think this one is pretty straightforward. I don't think it necessarily takes as much time and effort as some of the other ones. So that might be like a free bonus validation that you could do to win brownie points. So You might think most peptides are going to be stable, but we have unpublished, we have seen interesting modifications come out in some of these, and it totally depends on the amino acid sequence. Certain amino acids are more prone to being modified and under different conditions. So if you really care about your target a lot and you're really attached to your target, check it for these kind of stability issues. The last experiment is affectionately termed the 5x5. In my lab at least, and this is the one we dread having to do. It is, I think, the most work intensive, but it gives you such a good idea of how good your assay is. It gives you the best picture of how reproducible your assay is. So for this monster of an experiment, you will take your sample, a representative of your sample. So again, if we go back to that yeast example, I grow up a giant flask of yeast. Or if you're doing plasma, I've gone up to 2 liters so far. I'm hoping I never have to go that too much higher. If you're doing plasma, maybe you'll do a patient pool of plasma so that you have a bunch of different demographics represented in your pool. You'll aliquot it out so that these are unprocessed aliquots. You'll have 5 aliquots for each of 5 days. This is where the 5x5 comes into vernacular. Day 1, you pull out your 5 replicates of plasma, for example, process each of those 5. Day 2, pull out 5 replicates, process each of those 5, and onwards. The idea here is that you get a really good representation of the interday and intraday variation of your protocol. So one thing that might hugely influence the results of this experiment are the digestion kinetics. So Sue mentioned denaturing, but other kinetics that, other reaction details that can affect your digestion is just how much trypsin you're adding in, or how long you're doing that, or whether you're adding a bolus of trypsin at the end of a time. Desalting, if there's, for any strange reason, you're not getting reproducible desalt conditions. This should encompass everything. For me, one of my biggest sources of variation is the lysis. If you're working in plasma, I envy you because you don't have to worry about that. But if you're using cell lysates, lysis can be a huge source of variation. And this will give you an idea of where that variation is coming from. Is it coming from between days, or is it actually coming from the same day? Yep, so those are the key points that you're trying to get out of here. And I wanted to let you know you're not doing a bad job if you're eliminating targets. That means you're doing a good job. There is no shame in starting with for example, this is from the paper that we will be analyzing the data from. This group was interested in basically one single protein. So they started with 48 potential tryptic peptides to target, and they ended with 7. So there's nothing bad about that. They ended with the 7 most robust targets that they could possibly, and you notice they lost a peptide because the precision, the percent coefficient of variation, wasn't good enough. They lost 3 because they weren't stable when they did that freeze-thaw experiment. They lost another 2 because the signal wasn't good enough in the plasma when they did their response curves and they did those validation experiments. It wasn't a reproducible signal at the low end, and they ended with just 7. So you're not doing a bad job, you're doing a good job. So this paper, whose data we are going to spend the rest of this section thoroughly dissecting, was measurement of a vitamin D binding protein in serum, and they were comparing different methods that have been used to do this, and they ended up finding, interestingly enough, they proved their null hypothesis. So this is a negative data publication essentially. Other methods were reporting there was a difference between this depending on race of the patient. And this paper by Andy Hufnagel and one of his great staff scientists, Clark Henderson, they found that as opposed to what was previously reported in immunoassays, when they do this thorough, heavy vetting of a validated mass spec method, there was no difference in concentration depending on race. That was actually just really, really terrible quantitation on the immuno part. So it's a great paper. It's a pretty easy read because it's clinical chemistry. Highly recommend. It's a great example of doing these validations. So if you open up the Tuesday validation folder in the data, the flash drive data directory, you have to dig through a couple different folders to get down into where the actual data is, but you should reach a directory that lists 6 different directories inside of it that say bias, interferences, linearity, LLOQ, precision, and stability. Should be the 6 folders. If you go under the Tuesday folder on your data directory and drill down into that, you should see these 6. Everyone seeing these 6? Yeah? You doing good? Or at least on your way traveling to there? Any observations right off the bat? Remember I started with this soapbox announcement. Any observations right off the bat? I just got done telling you about these 5 experiments with very explicit names. Anything about these directories jump out at you?
There's 6.
There's 6, and I only talked about 5 experiments. So this CPTAC working group recommendation is a recommendation. It's not like a checklist necessarily. As long as you cover the key points they're trying to get at, that's what they're looking for, right? So I think I'm actually going to do this very differently than how we have done in the past. And I want to split you up into working groups and give each of your working groups one of these folders. And I want you to kind of explore the Skyline file in the folder that I give you. And then we're going to share, and then I'll give you a chance to look at all the other folders while you've got the 3 of us here and your peers to walk you through the data. Do you feel okay with the tools that we've given you so far for looking at data in Skyline? Or good enough to go out, branch out on your own and discover some more? So maybe 1, 2, 3, 4. Okay, let's do this front section, the 4 here, and I'll give you guys bias. So you can open up that folder and look at the bias. The 3rd row there, I'm going to have you guys in the 3rd row on the right side for you do interferences. Can I have the back row look at linearity? Can I have, you 4 together? You 2 are together? No, you can be now. Let's do 2 in the back, and the 2 gentlemen here do LLOQ. You can look at that. Let's do this side. If you guys can do precision, and then here do stability. And all I am looking for here is you guys just maybe open up the Skyline file, just get yourself oriented, talk amongst yourself, anything you're seeing in the data. Maybe take note of how many targets are on the left-hand side. Play around with the different pane views. Maybe you can open up the document grid. And then I'll give you guys, let's do 15 minutes to kind of look and just make some general notes, general observations about the data. I'll have you share with each other, and then I'll let you look at all of the data. And I'll be around if you guys are having problems or have questions. Yes? Oh, just whichever one. It's not super important. If you want to do either precision or stability. Just gives you a chance to, if I think a lot of us here have very different backgrounds. Some are very biology backgrounds, some are very analytical chemistry, some have a lot of Skyline experience, and some have no Skyline experience. It gives you a chance to be amongst the community and learn from each other, which is sometimes better than learning from an instructor. Yeah.
I'm just trying to figure out how to turn it on.
I think we're just supposed to explore.
This is ancient serum. This is that vitamin D.
Oh, the vitamin D.
Yep.
Okay.
Straight out. Oh, yeah.
So they look at
I think we can usually.
Across all these different.
You need differentiation. I mean, if you want to see all this, you have to keep in the one and you see all the and then you can go one by one.
Here's an actual was just on the first day. So what happened? So let's say. Yes, yes.
And then the next year,
we've tried to do a lot of different things.
there might be. For example, here is largest there's like and then the next stage, you get the stages of
How about your range of graduates?
yeah,
So these are
That was how you tell them to see all the independent guys. So, but at the end is we have to where they don't like it. Day 13, day 8 and 349. What's the I think so. When you're because I see 200 that one 230, right? 201.
Yeah.
And this is day 8.
Is there a guideline file or set up?
That's the light one. You see the bar. You see both. No, the point is that it's measuring
In the fact that says that it was fed into a FASTA file, that 295 is that sequence number of that even so they're trying to 302 is what that like so it's a position.
So in this case, one we need to
Better able to yeah, so if you look at these plots, the red signal is not based on yeah.
It's not. It's you choose a different type.
Yes, I think when we selected that part,
We open wide the window, so
we
it's
were
not
selecting that
that.
It's allowing you to pick like and peptide is two different so that's not really what we want to do with it.
yeah, so we can send it over here and go there too.
And then integrate
All of this data, how are you?
We were just selecting this part.
It's the order.
Oh, yeah, that's why. One is the correct.
So right now,
We even
It's a signal.
okay, and now we don't have the arrow here. We just have this. So the problem is how do you define say? You have to always be
Yeah, so what I try to do is before I have to go through this things, I try to run like a heavy light label. Yeah. Oh, cool. Like I have an actual in a context sample, and I get an idea of this. No, that's fine. That's kind of the and when you go to analyze. This is actual. This is you go to that data that this I'm not familiar with sample data. This is like this, and maybe this is actually covered. It looks like this is real life. This is peer reviewed and weird. Actually, so you can tell if you just did it by accident or not. Right click on the here. Yep. All right. So there, and then for that. in the directory, there is, and then, this is not, okay, so what's that, so that, that transition, and this is, beginning of the method, and those tag values, and yellow, and there's no, all the
It's a very risky. You can actually, at least
I always say, because if you have a experience in one of your transitions, how do you know which one is talking about, right, and to what extent? But you should do, like, if you have three, this is very there are people that do.
Let's check that.
Yeah, so no, I wouldn't ask, but what I would do is I would make sure that if whenever it goes to that the ratio of their responses their value get out. Okay, so because if it shows that results are totally but yeah, like we were talking about transitions earlier and all right, again, try typing all right. But try writing and the digestion rate something it's different. All right, so now we got AA. So unless you have a person that want to snap it to some area, this is without the having a diagnosis. But I would as a site probably rely on Yeah, so the way that this is set up is it's going to be integration. Okay, make any changes.
smart people like them. You know?
So the way this group sets it, oh, you got things something about it, everyone, okay.
Yeah, so the way that you had just had it when you had this, it looked like you might have to integrate it. I don't see it. And that's not the way to do it. You want to make sure that because there's a layer and every form they're going to go over it. So you want to integrate just around either reach or red. This one, the third one, right? I think it's fine like that. I don't know why you're maybe yeah, so I go
Less than
just zoom out from your view, and then F11 do it. But this is how the sample, this is how the data file. Right? And so that, but meaning that the blue one is the right key. That blue one is also the heavy, and that's something that that was additive. And so that means where you're gonna look for the length tag is gonna be right under that head. Oh, the line.
We get this is the data, this is data.
Yes.
And you calculate the
There should be a rate.
No. So adding the peptide to the sample.
I have leaking, but I don't know what.
Can you do me a favor, put on that
They just use testing of this.
It's the CPTAC. Are you guys all looking at the same freeze now? Yeah, the 1 in 3 minutes is the rate. So if you rate the box,
So the
and then go apply
I said, if this is bigger,
that's those are the data. Oh, yeah. Like the heavier clothes, you do that 3 minutes, and 1 in 3 minutes, you do it in minutes. So if you click on it, you just made it the right. You just made it right. So that's not the right one. Well, if you're doing a stability study, you're putting your light and your heavy peptide together, so there should be a light. Signal.
But I'm not gonna say that.
Yes, so it's a signal that peptide can generate. It's a Q1, Q3 transition that the instrument's monitoring. The instrument's not saying it's that peptide, it's just where these numbers and so that shows up later in the chromatogram that everyone has the same numeric value as your peptide, it's just not like it interferes. What's that?
Yeah, but look at it this one, maybe it's a pie, maybe it's
I think it's this mic. What?
Can I show you something? Okay. Yeah.
Why is there like more heavy peptide here? What do you mean by this? Because they study So the first thing would be, do you know
later stage? Yes, I guess
it's like
yeah, I think there's like a for that study. So that's one thing is if you looked up what those sample data meant, then you could draw more information from it. The other thing is if you right click in this window and you go online to then you can see the peak area ratio. So the light divided by the heavy, it drops down. For example, and then so you see the stable.
But the problem is that if you have to find out these things, you have to
prepare the samples, put them in the freezer, and then you can
so that might be the effect.
Call it whatever, and then once you place the bubbles in the same text, so they have different
It wasn't something I sent out. Okay. It might have been. I think it's on the.
So you have multiple like AAA or AAA.
That's all. I wanted to show you how to normalize. So the other thing.
There you go. I teach you
Okay.
So that would be a question that I would have to get this. I mean, they're like, I could do leaving explanations of maybe something is happening to factors that make it into, and now it's making it like maybe something is.
So now you're seeing all the statistics.
Well, if you look at it like.
I don't see that
Right, yeah, it looks, yeah, the light looks steep.
Because the difference you see because they
I don't think so because I know, yeah, maybe if you rate that, try it, it is not set. So what you would have to do is pretty straightforward. If you
it allows you to
you see that or not.
yes, or maybe the
So and then
Yeah.
so each one to are the same.
it's a red one. And then
So for me, I would probably
I guess so then if you were to set it in document settings, might have to add so click on
I am also a fan of subscribe.
and now it's arranged you want to group yeah, so if you do like A, B,
From 1 to 5.
like this.
Yep.
And with like this.
Oh, no, sorry, like A and then the next line A. Yeah, so call it first example the same day.
Okay,
And then
yeah.
And then now you should be able to go to your file, right?
I don't have to put
And so first, it's already set up, area is CP, but if you right click area apply, and you uncheck the CP values, it'll give you the address and the intermediation area. And then you go back. You can also like slide, look at that. If you wanted to do that, you would have to find that. And then show group by replicate, and then we have all of your replicates, and then you could calculate CB across all of those 6 replicates if that makes.
You guys maybe the last trip.
Why is that having higher in the free stock?
Right in the water.
It might be better in copies.
here, it sounds great.
Okay.
And then yeah, it's right now I need it in the second place and it is that's so cool apparently. There is a freeze, but we're gonna understand why. But what I heard is that means that there is no freeze in the light. So there is no so freeze those cycles in the even though even though the variance of the keep writing the variance, and then you have to Have you done the group by and now you do the C-value. I need to understand what the characterization is. How is calculated? How is calculating?
What's going on? So bias is
It's the variance.
you're always getting a little bit of scattering. So that's something because of variability. But you get enough.
Yeah, it's very much variable.
When we
Yeah, when I choose
Now, if it is
them, and then even if it's
it's because the I don't see something you would expect. Why do I have to turn this one in one direction?
you choose it.
I'm looking at the second one. I'd rather choose looking at the third Oh, sorry. You have to
Yeah, start first.
Yes. Result read. And then go to settings. No one No, you say okay.
What you say is heading
And then you
like that.
So I think what this is and then four and these four are different by when they're added.
Right.
And so they're addressing different points where variability can be introduced. Right? So if no variability should be introduced by and it's a variance, the question is how is that going to be and so one way we could do this.
But we don't have straight things. You can ask it to even in
So
a
y'all so there should be a buy option in there. If there's not, I can so I would also grab it would be like, all right, so everything goes to settings.
So
And then click on sample.
these means that the one that has more variances is no.
Now we present this.
Maybe it's just one thing.
Okay.
And now you see the samples there. So what you can do is like the it's a text string, you can make it. Yeah, if you do it with things numbers, it doesn't start the run. So I always do something like okay, good.
You will see that these go down like crazy. So I think that there's things like the dog example with cycles. Even though if you normalize.
So what you want to do is name the references from the same value. Now you area plot and right click. I will use this area plot the heavy is
Yeah, it's much higher.
so that's showing you your average area and then larger by standard.
So I think you have much more.
And again, that's showing carrier ratio. All of that. Like, someone that for some reason.
No, because
And then it's in the right. So they could carry and go to CPTAC. That shows you the variability.
some of them have
Yeah, so the ones that are very high, when I see a high one, the way that file is set up, I should go over and say, oh, because I have this one that uses the very like,
Yeah.
it's not gonna
you wouldn't do great.
So would you just take the average of this one?
Just choose it. Here's the
Yes. Yeah.
the first one is the not present. So that means okay, there is this the second one maybe and so now we can guess how much protein. So now it's that's why it wants to So in this case, we have to eliminate the first and fourth one. It's not there. And then we is just constant.
And I know like this one is going to be so I think this one is too
So you can still look at the signal that has variability by itself, so since the ratio is constant, when you get back, and it's like that. Because, you know, when you look at raw area, then that's a very key preliminary estimate. So you said it's a secret.
yes.
Not terrible. It's sort of
Yeah, but you will have a secret factor.
like, for example, your auto has to example. Here, as it was the whole direction.
Because what I see is
The ratio is good. But for the it's really
protein is going to be analyzed. And looking at
Now it's just has a full combination yeah. So now we have this one only not much presence like this one.
the right one is the right thing. The heavies are 56.
I don't know. It depends how, when you open the file,
And this one there is no bar over it. And that's in theory.
That's one of the 3.0 has a latency. If it was integrated, it was, yeah, it looks like it's not even there. But yeah, we can change the integrator. I don't want that. It is the so if
You don't know what.
whatever's going on at home is I don't know.
But you're right.
So let's see what I mean, I think if you that's the actual, it'll tell you everything in and then actual syrups in the lake. Was extremely low. No light. Yeah, so if we put 0.5, 0.5, yeah. Let's use those names next to each other.
But our purchase yeah, I would have next and so then maybe raise
Right? If you have synthetic peptides, then you can take the data from the calculator and calculate that peptide. Roll it up to the okay. Yeah. Those are easy if you have one side. But if you have two, they have to so that's replicated. Yeah.
you can add
But that's important to know, right?
yes. Even if it's like
I would so for example, if you compare all of our machines, if the response came back that we can group these two together as and that's like consistent from sample to sample. 75 is not correct because one saying I probably don't have all also
We're just, I guess, we made a lot of.
you already know for one peptide, and the one that's the most intense that you that is a cookie too. How would you? So the best you can do is calculate peptide concentration. I actually don't know this is not. And ideally, the way that I use it is I look at the concentration ratio I calculate from.
Well, we did, so
They all the samples are and ratio. So if it's always 2 to 1, and then if you look at
do you mean compared to the
no, like you actually have to label things.
okay.
And if one's ever they should always be that way.
Okay, in the old days.
And then when you start looking at different samples, if you look at then ideally you would see. So we always consider the same. So you're saying peptides should all, regardless of their okay. But yeah, it's tricky. I mean, but a lot of people are like, how did everybody?
designed to have your similarity in the center.
So they might have just been sure it doesn't matter to you guys.
Actually, this whole thing, big conclusion from this paper.
Same thing.
So this paper. Well, it failed to, well, yeah, so it's a negative data publication. So what they were trying to do, anti has like a little bit of a manifesto of destroying the entire narrative. As far as clinical Alexis, he
The rationale behind that set up by the they want to make sure that there's
and all of this work to perhaps
it is an abundant.
yeah. If there is a difference,
So that's why they just
There was a difference.
your question of completion, if that was more close.
It's nothing.
Yeah.
That's why it's very important. So that's why it's fine because the teacher writes can they also.
I don't, so yeah, I mean, and the idea is you want to make sure that you have a
This is the
good.
one for the this is thanks to the
so yeah, so this is like, for this experiment, for observation.
Well, the thing is that.
Okay, so
that's why it's
yeah, you get it. But that's why it's quantification, because you get it.
That is next. Yeah, we skipped the entire. Well, that's one because we're taking the whole effort.
So we said, responsibility, I don't have to say.
Oh, yeah, so doing that next.
it's not there. That's why you choose the yeah, they choose right one. And we not choose That's why.
Yeah, that part is right. I think.
Bigger space on the child. So that means the manual.
So you don't really want that data?
Yes.
That's one thing.
Yeah, that was good. Hopefully you got something out of it.
Oh, yeah. Well, just going into the system.
So that's 30% of one activity and there's like how you like do Did you find that validation
It's just including that.
time zero, time 4 hours, 24 hours. You lose sample, you lose
The first place that I'm using the little
You can see that error.
So I think we're just about ready for lunch break. I know I said we were gonna get back together and like all share our results, but it sounds like everyone found at least a couple new tools to put in their little Batman utility belt as far as using Skyline. From walking around, it seemed like some of the good ones for looking at this data were the sample group annotation feature. I encourage you to go on the Skyline site and look at all the other kind of annotations that you can use. Grouping the panes together and kind of just clicking and dragging and arranging them so that you can visually compare different chromatograms. What were some of the other things people liked? Anything in particular jump out at you besides those two? CV, looking at CV. I think Sue's going to be talking more about looking at percent coefficient of variation in Skyline using the group by feature. We mentioned normalize too. I don't know if you're going to do that later. Maybe we'll throw it in tomorrow into like the grab bag of extra things you can do in Skyline. Normalize too was helpful to some people. I talked to each other throughout the course about things that you're finding in Skyline or things you didn't realize you could do in Skyline, because the three of us have been doing this for a long time, so sometimes we forget what was that one cool thing that was so obvious to you guys that was so powerful that we just kind of took for granted. But have a great lunch when you come back. It's going to be calibration curves for days and days. So many calibration curves.